Welcome, everyone. Thank you for being here. My name is Crystal Hopper-Myers. I run our STEAM Pathways program here at the college. If you haven't been here to SWOCC before, welcome. So happy to see all the students in the audience.
Welcome, everyone.
Faculty members. Hold on.
Thank you for being
happen
here.
again last time. My name is Crystal Hopper-Myers. I run our STEAM Pathways program here at There we go. Sorry about that. It was one more thing. Like last time, it had happened the first time. We're like, oh no, we forgot to mute this. And like, apparently that happened again. So sorry about that. But again, I'm with STEAM Pathways. Basically, I just have this great opportunity to work with all these amazing faculty members, mostly in this health and science building right here. I welcome you to kind of walk around and check out the building if you have time. We have various different labs. We also have our awesome, on top of biology, geology, physics, chemistry, we've got our nursing faculty here, fire science, EMS program. So just please feel free to walk around and check out things. Like I was saying before, it's really great to see a lot of the faculty members and students out in the audience. And then of course our awesome community that comes out and supports these talks. If you have not signed in on the front, at the front there, please do so. It does help us kind of track how many people come and attend these lectures. So it's always helpful to kind of know what everybody's interested in and what are good days and good times to have these types. We've tried to kind of keep it consistently during this term. We have some awesome speakers lined up for our next term. I won't give away the surprise yet, and the fact that we're still working on a couple of them, just confirming them. I also like to give a shout out to, and we also have a couple of them in the, or a few of them in the audience. We have a great SPEAR student research team. SPEAR, if you didn't know what that is, it's STEAM Pathways, Experimental and Academic Research. Currently, we have a group of about 9 students that are working on a really cool project led by Dr. Wynn McLaughlin with a team of professors, because it's actually kind of a multidisciplinary project. And they are earthquakes, whales, tsunamis. They're just, there's a place called Fossil Point here. If you're familiar with the area, they're kind of doing some research out there. But these student researchers with this particular project have the opportunity to earn money while getting the hands-on experience. So it's a great opportunity for them to gain that valuable experience and kind of help get them a little money at the same time, because everybody knows if you're in college, it gets a little tough. So I know that because I'm still a college student. This particular award was a faculty research award given by Oregon NASA Space Grant Consortium. Dr. Coiner, who runs our physics and engineering department, he and I are local reps for this area. OSGC has been huge at supporting our students and giving them great opportunities like that. After completing projects like this, they become published authors. They get to present their research at the symposium at Oregon State. You all can come to ours we have actually a local symposium to see a lot of the research going on at the end of May, and we'll announce that to the community. So on top of OSGC, recently, we've actually connected with the Oregon Coast STEM Hub to provide high school STEM internships. So we've got some high schoolers that are actually part of that program. So with that said, I could talk about all kinds of things all the time about this because I'm just so excited about it. We do lots of K-12 and community outreach, but I'd like to hand over the microphone to Professor Amy Burgess. She is going to introduce our guest tonight. Thank you.
Don't want to stand in the projector light. Hello, my name is Amy Burgess. I'm one of the biology professors here. We're really excited to have Dr. Stephanie Carr here from Hartwick College. She is an associate professor of biology. She got her bachelor's degree from Hartwick College and then went on to get a PhD from the Colorado School of Mines, and then went back to Hartwick College. So Dr. Carr has done a lot of time at sea. She's done over 150 days of ship expeditions, and she studies the marine crustal biosphere, and excited to hear about it.
I have hopefully my own microphone that works. Well, thank you so much for having me here. And I want to also thank, like in the beginning, the National Science Foundation for funding my research, and also the U.S. Science Support Program, which funds people like me to come and give lectures on scientific ocean drilling. So I have a few goals, and I'm just gonna make sure this, here's me. One goal is to kind of tell you the history and the achievements of scientific ocean drilling. And then I'm going to tell you about how I culture with undergraduate students, the samples I've collected, and then how we can use our cultures to test some hypotheses about life in the marine crustal biosphere. Before I get into all that, I thought I'd introduce you to myself. So I'm from Maine, so like the other ocean, the Atlantic Ocean. And I just my mom had to like force me to leave the state. I kind of was a homebody and had indecision. So I didn't even knew what I wanted to do when I grew up. But I ended up at Hartwick College, which is not too far away, about 6 hours away from my family. And I did biochemistry because I always liked biology and I always liked chemistry, but I didn't know what to do. I knew I didn't want to be a doctor. I didn't want to be a nurse. I thought hospitals smelled. But like, what else do you do? I don't know. So it wasn't until I took a geology class and I realized that there's chemistry outside. When you guys have such great geology out here, and you see like the sedimentation and different colors. And I was like, oh, that's chemistry and elements. And that was really exciting for me. I wrote a first grant as an undergraduate student, which really developed my skills and was just like the start. Because if you're a scientist, you have to write about what you do a lot. So that was really important for me. And I did this grant that allowed me to do a project in petrology, so hard rock geology. And then I was like, oh, I do not like this either. So like, that's the important part about science. If you're going to school, like I know there's a lot of students here and you just don't know what to do. Like one good thing is to start ruling things out. I didn't want to be a doctor, didn't want to do petrology. I ended up going to the Colorado School of Mines because all my friends said, the rocks are in the west, so go west. And then I started working on Antarctic sediments. And that my professor there asked if I wanted to write another proposal to go to Antarctica and get my own sediments. And I thought, yeah, that'd be a great opportunity. So that was my first kind of taste of going to sea, and with a program called the International Ocean Drilling Program. So I'll talk about that more as we go on. From there, I did a small little year-long research assistantship. And this was more in biotechnology, and it was kind of biology, which is cool, but it wasn't outside. And so again, I rolled out and reminded myself like, no, I want to do stuff outside. And that's, again, just like keep trying things so you can like hone in on that thing you want to do. During that year, I knew I wanted to get back outside. So I started looking for grants I could write to find the money to support my dream. And so when I was like looking at, after you finish graduate school, sometimes if you don't go into industry, you do something that's called a postdoctorate. So I was looking for postdoctorate fellowships. And not only did I look for the fellowships themselves, but I looked for other people who were successful at getting money from those institutions. So usually they'll post like who else won awards. And I like looked at that list to see like who's winning the award. And I contacted those people and said like, would you be my mentor on these projects? And that was just so great to like reach out and find these mentors in the field. And so with that, I did a fellowship with a place called the Center for Dark Energy Biosphere Investigations, which sounds like something from Star Wars, like the dark energy. All that means is that I studied things that didn't do photosynthesis, and they got their energy from their environment and the elements in the environment. And then when I was doing that, their opportunities would come up where people would ask if I wanted to help them in the field. And this wouldn't be my project, it would just be me helping them. But I mean, first of all, it's fun. I really love going outside. I love going into the field. So I would always say yes. And then I started, looking back, I see that every time I apply my tools to a new situation, I become a better scientist. So, I mean, that's a great thing to collaborate with people, even if you're not getting the outcome yourself, like those aren't your samples or your paper. It was a great opportunity for me. So we sampled in Yellowstone. We sampled a place called the Dorado Outcrop, which is off the coast of Costa Rica. Some of the students were asking me like how I thought about deep sea mining. And it reminded me of this place because there's like so many cool ecosystems there that might need to be protected from places like deep sea mining. I did another fellowship, another National Science Foundation fellowship to kind of keep working in these environments. And from there, I got to go to places like a place called North Pond, which is like the Mid-Atlantic Ridge area. I worked off the coast of Japan, which is called the Nankai Trough. And then at the Bigelow for Ocean Sciences, they had a sea change program, which it was a lab. It wasn't a college, but they had a program where college kids came. And I remembered that I wanted to teach like somewhere along the line of my life. And that is like put the nail kind of in the board. I don't want to say coffin, but it was like, oh, wait a second. I had this epiphany, like I do want to teach. And so I was looking for jobs, and at Harvard College, they were looking for a microbiologist. And I thought, I want to teach at a place where I went to, like where I went to school. What better than where I went to school? So I liked it because there's undergraduate students working with professors, and and I really, really like that. So that's how I got back to Harvard College. So I just have like this big circle. But as I said, my whole career started with the scientific ocean drilling programs. And these programs, you might never have heard of them. There are these big collaborations where you have lots of different scientists. So geophysicists, geologists, biologists, hydrologists, geochemists, and they all come together on a ship. This is like my expedition to Antarctica. If you like look really closely, there's like icebergs in the back. And this is the picture of all the scientists and the drilling crew. And we're on these big ships. The US has had 2 drilling ships that are for science. The Goldmore Challenger, which was in the late 60s to early 80s. And then the Jody's Resolution, which came online in 1985. And you can see like there's this like big derrick. That's like where the drill pipe goes. There's like a hole in the middle of the boat, and they send the drill pipe down and they pull up the samples. And these samples, they come up as these big long cores that are 9 meters in length. So you can see that over here, we have this big long core, and we cut it into lengths, 1 meter sections, and we put them on this table. And what's really great about this program is that you have all these people collaborating, like different sciences coming together, different nationalities. This is an international program. And we all have different perspectives. And together we're trying to pick what are the best samples to be analyzed with the best instrumentation or the best tools to answer the questions that we want to answer. And each expedition has like a set of overall arching questions. And, if you can't, being there all together is like just the most amazing part of that, because something weird will come up and you'll be like, whoa, what is this in the core? And you can change your mind, and you can all work together to say like, wait a second, this would actually be the better way to go about this. I mean, people just stare at it for a long time. So it's been a really great program. And I say program, it's been called different things over time. like the funding kind of gets renewed and we give ourselves a new acronym, but and we kind of have different goals every time the program gets renewed. But it's been running for several iterations, and we just filed a report with the National Science Foundation, which helps fund it, which was really well received. And they kind of credit us for being the longest running and most successful international collaboration amongst the earth sciences, which just like warms my heart to be part of something that is thought of so highly. There's been 26 countries that have been working on these sites, and like over 5,000 scientific participants like are on the expeditions. So it's been this great thing. We drilled all over the country, I mean the world. So these are you can see the different colors represent the different years and the different acronyms for the science. But we sample like close to shore, which you can imagine has maybe high sedimentation rates, which is going to be really different from the middle of these like gyres, which will look really different. And we're kind of just studying the earth in all sorts of places. And you might kind of wonder to yourself, like, why do I care about the sediment that's at the bottom of the seafloor? But I promise you, you've actually have learned something from our science already. So if you've heard of seafloor spreading, like you have benefited from our science. I feel like I'm behind this thing. So the plate tectonics, that was a theory that existed for a long time, but it wasn't until we went out and drilled cores on the sides of the Mid-Atlantic Ridge did we get to verify the idea that it was spreading. So if you know about plate tectonics and seafloor spreading, you've benefited from scientific ocean drilling. If you think about the climate and how it is changing, most of our climate models integrate and are ground truth in science that came from scientific ocean drilling. This graph is from my research cruise to Antarctica, where we were really curious about what was happening around the Eocene, which was when the world was much, much warmer. And some of our questions were like, what was polar ice like at that time? How is sea level change going to change when the ice melts? And so we were kind of trying to think about, okay, if years are on the y-axis and temperatures on the x-axis, well, this is what it was like back then. Where are we going today? And the cool thing about it is, like, I relate it to a tree, because you know, if you cut a tree, you have tree rings. You can kind of like count the tree rings. You can see how old it is. You can learn things about the climate when you look at a tree ring. And the same is for the sediment. There's organisms that grow in the water column, like diatoms and dinoflagellates. And when they sink to the bottom, they hold secrets about the climate in their fossils. And so we can like look back at the layers and age when they used to exist and learn about those secrets. So that's how we kind of get our climate data. We can use our sites to study the earth budgets of earthquakes. So how they do that is we will drill a hole and we'll leave scientific instruments in there that can feel pressure changes. And when I told you I worked off the coast of Japan, while I was out there, there was actually an earthquake while we were on the drill ship. And we were able to pull up the instruments and then like see the pressure changes that happened in the seafloor. And they're looking at the earthquake itself, but then also, was there any signal before the earthquake that might like help predict earthquakes in the future? And so they're also like networking this in real time so they can watch those pressure changes happen in real time. We've also discovered that water flows through the ocean crust. So not a lot of people think about the ocean crust, right? Like it's below the sediment, but it's like this porous basaltic material. Water will sink into an outcrop, and then it kind of gets trapped there because the sediment is not as porous or permeable. And then the water will like travel and then come out somewhere else, like a hydrothermal vent. And that water is being warmed and it's touching the rock, and a lot of cool geochemistry happens. And so if we want to learn about the Earth's like global elemental cycles, the chemistry that's happening below the ocean is an important part of that. And then one of my favorite discoveries is that life lives in the seafloor. So these are some pictures. They don't show up great on the projector, but little life on basaltic rock. And then this graph here has depth on the y-axis and concentrations of cells on this x-axis. So you can see that life is decreasing the deeper you get. And this right here, this like plateau is not where life ends. It's just our limit of detection with the tools that we have. So we still really are trying to understand like how deep life can go. We're just like limited by our tools at this point, but it can go down. We see life like thousands, like 1,000 meters below the seafloor, which is crazy. So those are like some of our greatest hits. Those are some things that we have learned so far through scientific ocean drilling. And a group of us scientists have gotten together to make the new goals for the next 25 years. And we call this the 2050 framework. And I know these are in no particular order, but it does like bring me joy to see that number one is trying to see, to test the habitability of life on Earth, because I'm a microbiologist. So, you know, understanding the conditions that microbes need to live and what their role is in the marine biosphere. I'm excited to see that that is one of our 7 strategic objectives going forward. And we sent that to NSF, and NSF liked the plan a lot. So that's kind of like where we are now. We've had several different iterations of the program. We have a new plan for the future. We do have a little bit of a challenge because the JR, which is the Jody's Resolution, the US drill ship, has retired this year. So it's 45 years old, and that's about it for it. That I guess is how long drill ships last. So it has retired. There is a drill ship, like Japan has a drill ship, China has a new drill ship coming online. But now we are kind of shifting and trying to figure out, you know, what do we do with the samples we have? How can we use other platforms to continue drilling? Can we get another drill ship? We're looking into all these things. Another thing is that it's expensive to run this program. It keeps getting more expensive, and it's hard to get the international collaborators to keep up with that expense too. So the NSF has asked us, like, well, how are we going to deal with that cost? So we have a bunch of us like trying to think about that. There's a working group called the Future of Ocean Drilling in the US, or FOCUS. They have like lots of ways that the community can put input into these decisions. So if you're interested, you can look that up. Okay, so that's an awkward transition for scientific ocean drilling, and it's an awkward transition in my talk. So I'm switching from all our greatest hits to what I specifically studied. And to do that, I want to introduce you to the Juan de Fuca ridge flank, which is not too far from here. So it's off the coast of, you know, Oregon, a little north, closer to Vancouver Island. I have had the pleasure of going there twice, in 2019 and 2023. And we left from Newport. So people have asked, this is my first time in Oregon. It's not, because I've got to go to Newport a few times. At this site, so there we took the Jody's Resolution and we drilled into the seafloor, and we left behind what we call these subsurface observatories, these borehole observatories. It's very similar to a well if you have well water on the continent. Basically, we've just left, we kind of sealed it off. Sometimes I call it a cork, like you cork a bottle of wine. And so that the seawater can't get back in, and can't contaminate the subsurface. And we just have umbilicals that go into the crust, and we can use that to pump out the water and collect the water. Sometimes we leave instruments at the top of the wellhead. So this is like a photograph of one of the borehole observatories. These milk crates hold like little pumps that are slowly pumping the water and sampling it. And we can leave it there for a few years and then come back and get those samples. Other times we take an ROV and we just plug into the cork or the observatory. And that's kind of like, we do that at the top where these like yellow horizontal bars are. And the ROV has some power from the ship, and we can pump a lot of water at that point. So me and my collaborators, Mike Rapay and Olivia Nigro, we wrote a grant to go back to the Juan de Fuca Ridge to do some science. A collaborator also wrote a different grant to go back, and we shared the ship time And so Beth Orcutt was that collaborator. And we went out in 2019 and then again in 2023 to do some great science. I was lucky that in 2023, I got to be the co-chief, which was my first time being co-chief, with co-chief, I should say, with Beth Orcutt. And like one really cool thing about being co-chief is you get to help pick the team that goes on the boat with you, the ship with you. So there's only so many berths, and you want to make sure you pick the right collection of people that are going to get the science done. It was really important to us that we were raising the next generation of scientists. So we really invested a lot of this berth space to students. So we had 5 undergraduate students, 5 graduate students. We had a science educator, which was also a student from my school. Our biggest teammate is Jason, the ROV, which is in the back. And Jason comes with its own team of people. So every time Jason goes out to sea, it goes with 10 people that takes turns driving Jason, because Jason can operate 24/7. So they work in teams 24 hours a day. And then we also had a training student for Jason too, because you got to train new Jason pilots. Another really cool thing about this expedition is that the place where the Juan de Fuca Ridge quarks are we knew was going to be classified as a marine protected area. And that classification was coming about, it's in Canadian waters, so it's partly the Canadian government, but they were working together with First Nations people to make this area a protected area. So it didn't actually happen until 2024, but when we were planning this in 2023, that was something on our mind. So we also brought 2 First Nation observers just because we wanted to make sure that we were sharing our science with their community and also gaining their perspectives. And that we want, and I hope that is a precedent going forward, that if we're going to other people's lands or places that are protected, that we include them. And so the name I've been practicing for a long time of the marine protected area is the Tangwan Hachquikuk Tigis Marine Protected Area. I maybe did the best I could. And it is 3 different languages pieced together that basically translates to the deep ocean with monsters. And so it's just like great having that whole team together. The education officer, as I said, was actually an education student from Hartwick College that is they're doing an education certification as a biology major. And so this is Andrew DeCernia. He was he is still the education officer. Before we went, we talked to 7th grade classrooms in our town. We did 5 library events. We did an art project where kids drew on cups, styrofoam cups, and then we bring them to the bottom of the seafloor and they shrink, and then we return them to the library and the kids could pick them up. And then he's also helping me develop an undergraduate activity for the bio program at my school. And we're presenting that at a conference this week, actually, on Saturday. So it's been really fun working with Andrew. So that's kind of like some of the outreach we do. We also do science. So our grant has 3 goals. One was just to kind of continue the long-term sampling of these sites. So we've been going there for decades, and we just want to continue the science there, continue counting how many cells live there, and then getting their genetic material, and we can use their genomes to learn what they're doing. We also wanted to isolate novel organisms. So from the genomes we've been collecting before, we know that there's some novel organisms that live there that nobody has cultured before. So that's a big goal of ours. And that's the goal I worked on the most. And then also Olivia was there to kind of identify and isolate new viruses. I didn't work on the viruses, but we did think about them. So I wanted to ask the students, if you were a biologist, or even if you're not a biologist, and you were supposed to culture something from a new environment, like what would you need to know? Maybe you weren't thinking someone would ask you a question. Oh, and maybe I didn't prepare. Anyone with the mic? Does anyone have any thoughts of like what you might want to know? You're trying to grow something new. Yeah, you want to know what it's made out of. That's good. And because if you knew what it was made out of, you might know what to feed it. Is that what you would get to? Yeah. And like, what else? If you were growing a plant, what do you need to know about the plant? Yeah. What kind of soil? What kind of sunlight? Yeah, these are great ideas. So yeah, that you need to know the environment. Good. So that's, if I tell you about the environment, it's warm. It's 60 degrees Celsius, which is like 140 degrees Fahrenheit. So that's pretty warm. There's no oxygen. So these things don't breathe oxygen. They breathe other chemicals. A lot of them use sulfate as their electron acceptor. And there's really limited carbon. So yeah, I know that they need to make lipids. I know they need to make sugars. Like, where are they getting those carbon sources? But I do know there's a little bit of organic carbon down there. So we've taken all this ideas. The other thing we do know is we kind of know who we're looking for. So this is like a ridiculously small graph, and you don't need to read any of it. So you're just going to focus on the colors, and I'll walk you through them. So we have a few of these borehole observatories that are not brand new, they're newer. And they were made with non-corrosive materials, which is better for microbiology. So they, like Teflon, for example. And those observatories are highlighted in this pink and red color. And then these are the different types of organisms that were found. And we have other sites too. So this blue color is just bottom seawater. This brown color that's not showing up really well is the sediment. And then this yellow color is a leaky observatory. So it's a place where the seal didn't work quite right. And so it's like subsurface water, but also ocean water mixing together. And so what this graph is showing us is that the microbes that are coming from the well-installed new technology observatories are really unique. They're not the same as the organisms that are at the bottom of the seafloor. They're not the same as the microbes that are at the sediment. And they're, this yellow is kind of like a mix of what is found in the seawater and what is found in the borehole ones. So we're pretty confident that our new observatories give us the subsurface environment. And what we find in those observatories, when we look at the genomes, we find things like Hydrothermarchiota, Aminocyantias, Archiglobus. And these organisms, when we look into their genomes, we see things like they use sulfate as an electron acceptor. So they reduce sulfate instead of reducing oxygen. Some of them are chemotrophs, which means they can use like CO2 to make their lipids and their sugars. Some of them are actually heterotrophs, even though there's limited carbon down there. Some of them are both. So I kind of use that information to predict what I would need to feed these microbes. And then we made different media types, and we have to put these special stoppers in here to keep the oxygen out, and we flush them with nitrogen or hydrogen sometimes. And this is my student, Sierra Brown. She's my biology student. This is Andrew, the educator, who is getting his hands dirty and like doing the science too. This is Jason, our ROV that goes to the bottom of the seafloor. This is us in Newport. This is the Thompson, which is the research vessel we took. And so the ROV Jason goes to the bottom of the seafloor, plugs into the observatory, and then brings the water back up in these like aluminum bags that are hopefully keeping the oxygen out. And we have all this technical equipment, and then what we end up doing is stacking boxes on top of each other, and the water is in the top box, and we flow it into a nitrogen gas bag so that we keep the oxygen out, and then we can use it to put it into these vials that have the stoppers. So that's our big goal: keep oxygen out. And so we tried 11 different media types. We tried feeding them different things. Media is what you feed them. And I come from a school that's just undergrad, so there's no graduate students. And I've learned a few things about culturing. In 2019, we went out and we collected these samples in a very similar method. One thing we use to usually look at microbes is it's a measurement called turbidity. So turbidity just means like cloudiness. And so if you have like clear water and you put a microbe in there, it will like usually multiply and become really cloudy. Also, though, I learned that if I make microbe food media in the lab, and then I throw in some crustal fluid, sometimes chemistry just happens and you get a precipitate anyway. So it's hard to tell if it's really life or if it's just chemistry gone wrong, wild. A better way to look for life is to do microscopy and to stain the cells. But what I learned with my undergrads is that takes way too much time. If my undergrads are taking classes all day, which they are, it takes a long time to prep these slides, and then you can get lost in the microscope for hours, like looking for cells. So that was hard. I also learned that pandemics are no good. Like they sent the students home, and if I had these dreams that these students were going to grow microbes continuously for me, like that dream was dead. So it was a hard 2020 for my microbes and me. But I did learn that I could measure life by measuring ATP. So ATP is just like this energy molecule. And I learned that the students can do it in 5 minutes as opposed to doing microscopy for hours. They could do several at a time. And it took like very little sample to measure, just like a few drops. And microscopy takes milliliters to do. So this was like the ticket. This is one of my students that actually was with me. Well, she started school during the pandemic and she helped me afterwards. And Tylisha over there went on the 2019 cruise and then was sadly sent home during the pandemic. This is our instrument that we measure ATP on. Now we have actually a plate reader that does it, and I can measure 96 at a time, which is great. So I had this new strategy for 2023, and we took our 11 media types. So this is like all the different types of food that we tried to grow the microbes on. And so I have some that are heterotroph, meaning I fed them organic food. Sometimes I tried like feeding them just lipids or feeding them just amino acids. I tried feeding them CO2. That's what autotrophs mean. We gave them different electron acceptors. So I gave them sulfate and nitrate, and we gave them all these things. And so these are the ATP measurements. And what we were going for is we were just trying to see if we could grow anything that produced more ATP than the blanks. And so the blanks are represented by this red line. And so any dot that is above the red line means it had more ATP than the blank. And our goal was just to like grow one thing. And we grew like so many things. It was kind of overwhelming. Some of these green ones means that like it was a co-culture. So that means sometimes I thought that maybe a microbe would grow better with a friend. So I not only made the food to feed them, but I gave them a little friend. And so the green is the friend alone. So you can see here, these ones didn't do much better than the friend by itself. And maybe that was just the friend getting like extra excited when we put in the crustal fluid. So, but we had a lot to go on, a lot of samples that showed more ATP. So that means we grew what we call enrichments. So we grew communities of microbes from the deep biosphere, which is an excess upon itself. And then we wanted to isolate organisms, so get them all by themselves. Because remember, nobody had done that from this place. So we wanted to do that. And so this is Sierra. And a lot of times in microbiology, I know you have a microbiology lab here, you grow isolates on agar plates, like streak them out. And the agar plates are just kind of like Jell-O. You see little dots, but like Jell-O, it melts at a high temperature. So we can't do agar plates if we're going to grow things at 140 degrees Fahrenheit. So we have to just dilute the samples until we have only one cell at the dilution. So what my students will do is she comes in the morning, this is Sarah, she'll come in the morning, she'll grow a culture on one of the enrichments overnight. She does microscopy and she quickly counts the cells, which is, has a little bit of error to it when we try to do it real fast. And then she'll make several dilutions, and then she'll make some more media, and we get a 96 well plate, and she puts the controls in a few of the wells so we know like what media without cells looks like. And then I find it best to try different dilutions on the plate, because when you're counting cells real fast, like we don't have all day to make sure that we have the count just right. But we put 3 different dilutions on one plate, and we have this goal that if to get just like 1 cell in a well. And so our goal is actually to get less than 1 well in a cell, so that we're sure that when one lights up, that it's a real isolate. So we hope in this middle concentration that we get the dilution right, but we do have like a little wiggle room. And then she can measure the ATP in all 96 wells, because we have a 96 well plate reader, and see if she was successful at isolating anything in the wells. And so what she has been focusing on is the heterotrophs with amino acids. And then she also did this Archiglobus media, because remember, we knew that there was Archiglobus, and Archiglobus has been grown before from other locations. And what she's been able to pull out is we have 1 Archiglobus isolate and 5 of these Cosmatoga isolates. And so now we can go into these isolates and start characterizing them, which will be fun, but we're just starting to get the isolates, which is great. So that is culturing with undergrads. And then this next thing is kind of like a cooking show. Like when I put in the oven and then take out an idea. So we have the culturing, which is happening right now, where we're isolating things. And then the next part of that is doing something with those isolates and like testing hypotheses. So I'll show you how we can test hypotheses, which we have hypotheses for Archiglobus, even though we're using strains that we bought, but I'll just give you an example of that. And when I started at Hartwick College, people were really interested in my research, because they were like, wow, this is a new environment we've never thought about. And one of my colleagues that does molecular biology asked me about riboswitches. And because I am more of a geochemist, I didn't really know what a riboswitch was. I'm imagining you might not know what a riboswitch is. So I'll tell you about them first, and then I'll tell you my hypothesis about them. This is a riboswitch. So riboswitches are segments of RNA. And I'm going to assume that you might remember from biology that RNA gets transcribed into DNA and gets translated into proteins. But RNA is single-stranded. And so this RNA, it's single-stranded, and it folds upon itself in a way that it has these interesting loops. And sometimes the big loops will have an impact on the downstream transcription or translation. In this case, I'm showing you an impact on transcription. So if this is RNA polymerase, which is doing the transcription, which is making DNA, this big loop is actually a terminator loop that kicks off RNA polymerase. So there's no transcription, and then therefore there's no translation, there's no protein. So this configuration is stopping something from happening. If there is a ligand that matches the riboswitch, it will change the conformation of that RNA so that the terminator loop is now smaller. It allows RNA polymerase to continue, DNA will be made, and then translation and proteins will, translation will happen and proteins will be made. So it's really interesting because it's a way that a microbe's genome is kind of impacted by an environmental signal, like something that's in the environment. So that's what a riboswitch is. And I thought they were cool because, yeah, it has this connection between the environment and then what happens to the cell, like the physiology of the cell. And they're also kind of cool because it is thought that they might have come from an RNA world. So before DNA kind of developed, maybe life was just RNA, and RNA had to be both the genetic code and also the enzymes that did everything. But they're not enzymes because enzymes are protein, but you know what there would be before enzymes. And it's also really interesting. My colleague was interested in it because riboswitches could be cool for biotechnology. If you were thinking you had to make a product and you were training a microbe to do that reaction, and you could turn that reaction on and off just by adding fluoride, for example, right? So these are all cool reasons why we wanted to look for riboswitches. And he was thinking, because we have this really unique environment and this really unique set of microbes, maybe they would have unique riboswitches as well. So I had a student, I'll show you her picture in a minute. And we looked for riboswitches in our genomes. And it turned out that the most abundant riboswitch was the fluoride riboswitch. And then I thought to myself, like, well, I wonder how prevalent riboswitches are, the fluoride riboswitch in archaea. So in the tree of life, there's eukaryotes and bacteria and archaea, and archaea is understudied, especially in the world of riboswitches. So we were going to do that. And our hypothesis was that if riboswitches evolved early, like this RNA world hypothesis, that they would be throughout the tree of archaea. And so that's what she was looking for. She was looking in all of these archaea genomes, and she was looking for these riboswitches. Oh, this picture doesn't look as great as it does on my screen, but that's okay. So our expected results, again, we were looking for where riboswitch might be in the tree. And what I have here is this color codedness, where the pink and like orange colors represent higher amounts of organisms within a clade that have that riboswitch. So, and then the gray and blue represent the lower amounts of organisms within that clade that have a riboswitch. And so what we found was she looked at 4,000 genomes, over 4,000 genomes, and she found that 700 of them had a riboswitch. And that those riboswitches are kind of concentrated, the oranges and pink are concentrated in the top part of the tree, and not in this bottom part of the tree, which I kind of refer to as the depan section of the tree. But there's still kind of like there's some in most branches, which gives us this kind of conclusion that either riboswitches did evolve early, but they've been lost through evolution, or they came about many times through evolution, which is less likely. So we think we have support that these riboswitches are an early mechanism that has just been lost in time. And we kind of see that this looks better. You can kind of see that like in this group, Methanobacteriota, within that clade, there's many organisms, and 29% of them have the riboswitch. And if I like dig into this, so this tree is phylum. And if you might think about like there was phylum, order, class, family, species, genus, right? So if I dig into the phylum and look at the orders, I can see that maybe it was lost at this point of the tree, but stayed within that class of Methanobacteria. So you can see like points in the tree where maybe the riboswitch was lost. The other thing that people can do is they not only look at the riboswitch, but what the riboswitch regulates. And very often, 30% of the time, the riboswitch regulates a gene that is a pump that then pumps out fluoride, which kind of makes sense. So like if there's fluoride present, this configuration allows transcription to happen. It transcribes a gene for a pump that then will translate to a protein, which will pump fluoride out, because the cell doesn't really have a lot of need for fluoride. But if there's not a lot of fluoride, then you would turn that off and you wouldn't bother making that pump. So, I mean, that kind of all makes sense. And what's interesting is that when I made a phylogenetic tree for those pumps, a lot of the pumps at the bottom of the tree actually they were more related to the organisms within Halobacteriota, which kind of makes me think that maybe this whole branch, what I call the depan branch, lost the riboswitches, but then has gained those riboswitches back through a process called horizontal gene transfer. So that's my running hypothesis with that now, but we're still diving into that. What's interesting though is not that these pumps are only follow the riboswitch 30% of the time. So then you have to think about like, what is going on with the rest of the riboswitches? Do these genes that follow the riboswitches, do they always kind of, are they inherited with the riboswitch all the time? It's very complicated. So we're kind of diving into that. And I have an example within that Archiglobus family, species, sorry, genus, oh my gosh, that we have 3 species within this genus. They all have a fluoride riboswitch, but they don't have the same things that follow them. And so the first one, Archiglobus cephalicalis, it does have that fluoride pump. So that is like, okay, I kind of understand that. The Archiglobus fulgidus, it doesn't have specifically a fluoride pump, but it does seem to have an ion channel, and fluoride's an ion, so maybe it's using that to kind of pump out fluoride. And then Archiglobus neptunus doesn't have anything after the fluoride riboswitch, except for this prenyltransferase, and no one really knows what that's doing, but it seems to be in all 3, so maybe it's important. So my student and I had this hypothesis that maybe ideally it's best to have a pump after the riboswitch, and that we had a hypothesis that if we tried to grow Neptunus with fluoride, that it wouldn't grow as well as the others, that the pumps of cephalicalis and fulgidus that helps those strains grow with fluoride. And for whatever reason, the pump was lost here, and that strain might not grow anymore. And we were just very interested to see like how what exactly is being regulated by these switches, so that if we wanted to apply this to biotechnology later, that we would understand these switches a little bit more and how their regulation works upon other genes. So we designed an experiment. And this it started with Cha Akrasan, and she graduated. So now I have Jadon Demerit and Jack Preston that have been working with me on this project last summer. And we've been growing the 3 strains of Archiglobus under different concentrations of fluoride. And we've been counting them. So we filter the cells and we put them under the microscope and we count them. We also measure turbidity. I'll show you those graphs too. And then we extract the RNA to see under what conditions, what genes are transcribed. And these are our graphs, our growth graphs. So this is the turbidity, because when we do the microscopy, we end up sacrificing the whole culture. So as the cells grow, I told you they kind of get cloudy. And so that cloudiness increases with time. That's what these graphs are showing. So with the cephalicalis and the fulgidus, we underneath the different concentrations of fluoride, it's almost the same growth. So it seems to me that those pumps are helping those cells grow in the presence of fluoride. With the Neptunus, we tried to grow it at the highest concentration of fluoride, at the 5, which in this graph is green, and it didn't grow at all. So then we lowered it to 4.5, so now we have this new color orange. And you can see the orange is growing, but it's still not growing as good. So it seems to be struggling when we add a lot of fluoride, and we kind of have this hypothesis is because it doesn't have a pump to help get that stuff out. So that's what I'm highlighting here. You can see the orange is less turbid, it's not growing as good. What's really curious though, isn't it, with enough time, the orange does start to overcome the other concentrations, which we think is really cool. And we've done this several times and we keep seeing it. So we actually did the experiment one last time where we can measure our RNA and cell counts here and here. And all these other graphs, we just sacrificed them once because we didn't know we were going to be interested in 2 time points. So that's what the students are working on. And we are at the point where we have extracted RNA and we're starting to run these analyses, which are called qPCR, which just tells you what genes are turned on, basically. And we don't have them all done, all the runs, but I can show you a little bit of those results. So if qPCR, the way we look at those results is called relative quantification. So we have controls, and in our case, the control is when there's no fluoride. So just normal cells growing normally. And now remember, if we throw in fluoride, we hypothesize that the pumps are going to be turned on, or whatever is next to the fluoride riboswitch will be turned on. So if that is the case, if a target gene is turned on, then the result is going to be more than the control. So if that's going to be like, if the genes are turned on, it's going to be greater than 1. If the result is less than 1, that means the target gene was actually suppressed by the fluoride. And if it's the same, if it's 0, or I'm sorry, if it is 1, then that means there was no change. So Cha started this last summer, and we're starting to repeat her results. We haven't got that far. And we've only tested the parental transferase because it was easy, because they're in all the strains. And so when we added fluoride to the Archiglobus fulgidus, we saw that the next gene was turned on, because it's a 77, so it's much greater than 1. So it seems like the fluoride riboswitch is working, and it's regulating the downstream genes, which is perfect. In the A. nuttunus, it was actually almost 1. It wasn't like that much greater than 1. It's not much lower than 1. It's actually almost the same. And that was interesting to us, because it means that fluoride, there's something about this fluoride riboswitch which isn't working, which goes with our results on the growth curve, which showed that microbe was struggling to grow with the fluoride. And I thought it was because there was no pump, but it seems to me that this riboswitch just might not be working at all. And it's really, when we dove back into those genomes, this picture, actually, I try to show it. There's a little bit of a gap in between the fluoride riboswitch and the parental transferase and the fluoride riboswitch and this like unknown, uncharacterized DUF 190. But here there's an overlap. So the riboswitch actually, the code is kind of compromised. And we think that is why that microbe isn't growing that well. So we still have a lot of qPCR to do, because we want to look at all of the expressions of these genes, but we're just getting started. So with that, I guess I'll just summarize what we've done. Like we looked at, I hope I introduced you to scientific ocean drilling. I hope you think that it's a valuable program. And even if we don't have a drill ship right now, we're going to still be pumping out great science. We are growing new isolates from our borehole observatories. And what's nice about the borehole observatories is we don't need a drill ship to go back to those places. We can go back with smaller ships and the ROVs. And then once we have those isolates, we can test hypotheses like, how do riboswitches work, and how can we make sure that they're working? So if we do want to apply those to other technologies, that we can get that right and understand a little bit of how they evolved and what that might mean for early life on this planet and what it might mean for life elsewhere. And with that, I'll take any questions. Thank you for coming.
I'm going to check in with our online listeners as well, so in case they have questions, but if you would just raise your hand, I can bring the microphone to you.
See that back.
Does age of seafloor sediments impact like the amount of fluoride and various things like that?
Yeah, that's a great question. So I do not know the answer to the age, but I do know that fluoride is really abundant in the crust. It's the 13th most abundant element in the crust. It's at it's like at a pretty significant concentration. Another person asked me, which I thought was interesting, is maybe the loss of the fluoride riboswitch kind of also maybe happened at a similar time where there was less volcanic activity. So I am really interested in that question of age, but I haven't dove into it yet.
How do you count like the cells? Do you literally just put it under a microscope and then just count it by hand?
Yeah, yeah.
Oh, that's all.
Yeah, I mean, it's very interesting. So my colleague kind of described counting cells as like looking for pennies on the bottom of a pool that is also painted copper, which is not as bad as it really is. But you stain the cells, and the stains usually bind to DNA, and then you can count the cells. But the stains also bind to other things. So you also have to kind of decipher like, oh, that's a cell and that's not a cell. So it's tricky because it would be great if I just had a computer program and taught AI, to count everything that's green. Yeah. But I also have to be like, well, nothing too big because that is not a cell, and nothing too rectangular. It has to be circular. So there's a lot of we do kind of do that, but it takes also human verification to make sure that the computer is not counting things that are not cells. Oh yeah. So yeah, so that's why we count them early in the morning. We get a quick estimate, and my students have class, so they have something else to do. So we do this quick count, and then we just dilute it. So it's usually they don't grow to terribly high concentrations. So they might grow to times 10 to the 6th or times 10 to the 4th. And then we just calculate that dilution series. But you're right, if our count is bad, then our dilution will be bad, which is why I do 3 dilutions on the plate so that there's that wiggle room. So if we were just way off, then hopefully I'm got 2 orders of magnitude on both sides. So hopefully I get it. And then we're aiming for like 0.3 cells per mil. So we're hoping that most of the cells are actually empty, and so we're like trying to underscore it so that when we do have one, we can be more confident that it was all alone. Thanks. And then when we grow it, we look at it under the microscope and make sure that it looks at least the same morphology. And then we'll do some DNA to also make sure that it seems like it's just one genome.
Your talk was divided into 2 parts. I have a question for each part.
Okay.
The first part was the basic international deep sea drilling project. Assuming magic happened and there was unlimited funding, what is the single most important research topic that should be addressed?
Oh my gosh. There's so many. It's so hard to pick one. That's why we work together. I do the microbiology, which I think is so cool. If you think about life on this planet and you think about all the trees you see out there that are living, all the grasses, all the plankton and seaweed, you add that all up, like the same amount of life is underground, and we don't even know like what it's doing and what we could harness from that. So in some ways I love that. But I also think that as a human society, climate change is really on my mind all the time. And so to keep modeling that is really interesting to me. And I know. So I also think that is really important, and just making sure that, but I don't know, that we can try to predict what's going to happen. But it's happening. So.
Okay. The second question, more regarding to your experience with these crustal things. We live in the realm of Cascadia, and which means there's subduction going on, and water, which contains these things, is being carried deeper and deeper and deeper. The temperatures are changing. Things are happening as it gets deeper. And a lot of times this water ends up coming up here in volcanoes.
Right.
What happens to the microbes as the subduction plate keeps moving down further and the environment, the temperature changes? What happens?
Yeah, that's a good question. And my colleague, Karen Lloyd, she is really interested in the microbes that are going down into the subduction zone. She does work in Costa Rica. areas like that. I mean, the communities, I think they'll change, right? There's microbes that won't be able to tolerate those higher temperatures. I mean, if they all get into the mantle, I don't think they'll come back up the same. Something else is going to come up. Like as warm water comes up, it'll collect things and bring it up. That's fine. I don't think that microbes are going to survive the mantle if they get down in there.
Anybody else have any questions?
What type of cells like are you like I know they can be plant cells because there's no sun, but like is it like animal bacteria? Like what type of cells are you looking at and studying?
Yeah, so they are the ones that I'm looking at, this high temperature environment is mostly archaeal. So, but I should say the Cosmatoga ones that we have been successful at pulling out are bacterial. The Argiglobus is archaeal. And I think I think that the Cosmatoga ones just love the amino acids we're feeding them. So that's why we keep pulling out the same one from our hat. If it's like a rabbit, you're pulling out of a hat. What's cool is they're slightly different strains. So I'm interested in seeing how much variability there is in the environment. But yeah, it is an interesting site. And this site, the Juan de Fuca site, is warm. But there are other parts of the crustal biosphere that are cold. So if you went to the mid-Atlantic ridge, that area, the crust there is actually colder and there's oxygen. So the microbes are totally different. And then what's crazy is that those are the two places we have studied. And there's a whole lot of ocean, so we have basically two endpoints. And there are endpoints, they're not necessarily the endpoints, where we have a hot environment where there's no oxygen and a cold environment where there is oxygen. And we don't really have any other sites that we study the microbiome of that taps into that crust. There's also continental crust. So I do have a project, I'll show it to you. I have another slide where we have this is at the Cornell campus, which is really convenient for me being in New York. They just drilled a 2-mile observatory on their campus. The main driver of that was geothermal energy, but they're also interested in what is living down there. And it's really briny down there, which is another cool analog if you're thinking of life on other planets. It's also really warm. So it's about 80 degrees Celsius. So it's warmer than the Juan de Fuca site I study. And there's Sierra. You might recognize her from the poster. There she is. And some other students that I brought down there, and we are collecting from there. But there's very few sites where we can tap into the crust and look at these microbes. So these also are turning out to be bacteria and archaeal. I haven't really tapped into I haven't looked at eukaryotic things because they have a different genomic signature anyway, but I have a colleague that is looking at those, and we have a colleague that's looking at viruses. Thanks.
When I think of microbes, I think of communities and interactions amongst them. What's known about the microbes? Can you tell us about how the cells make living?
Yeah.
What about symbiotes?
How they live together. Yeah. Yeah. And I wish I had more to tell you, but I'll show you this picture again where we have this idea that these microbes would love living with a friend, right? So that they would work together. And it's not so over here, it didn't work out maybe as well, but we do have this over here, we grew them with methanogens, which are microbes that make methane, but they do that. They're fed by other microbes to make that methane. And then over here, we grew them with Archiglobus, which we knew was in the environment. And my other collaborators designed those media, but we're working to pull things out of those as well. But I do think that they probably work together. And what's really interesting about the crustal biosphere is that there's a lot of microbes with what I would call like an appendage, which you would think it's kind of like a flagellum, which maybe would help them swim around, which might be true what they're doing in an environment that's like flowing. But I also think that these things, I think, and it has been shown that they can be used to like attach, and they'll attach to each other and grow biofilm, or they'll attach to rock. It's hard to pull up a pristine rock and look at it. The drill just like tears everything up in this rocky environment. Like in the sediment, you can kind of like push, like you do with a milkshake. You put your straw in a milkshake and you put your finger on it and you can pull up the straw, and then there's like the milkshake is on the straw. So that works out really good for the sediment, but it's not really, it's not what happens in the crust. It like grinds it all up. But to think that they're making biofilms on the rock, and we do have like some of the small samples we pull up, it does look like they can make biofilms on the rock and form a biofilm where they can work together and share things. So if, for example, one is eating an amino acid and they break it down to CO2, then they could feed that methanogen, and that methanogen would live its life. So I do think that's happening. And so continuing to pull these things out, and then it'd be great to grow them apart and then grow them together so you can see that happening. And so that's kind of why we're trying to isolate them. But even if we don't get those isolates and we just get co-cultures, we can try to look at them together. Yeah.
It doesn't look like we have any questions online. Does anybody else have questions here? Okay. Well, thank you,
Yeah,
Dr.
thank you
Carr,
for
and thank
coming.
you all for coming tonight. We really appreciate you all coming out, and again, look forward to the next term of lectures. We will announce them on the website at SWOCC.edu. As you can see, we live stream them on YouTube if you don't get a chance to come out with like weather or if you're ill. And then but we have a nice lineup. And if you're on Facebook also, we stay active on the SWOCC STEAM Pathways Facebook page. And they also try to get it out to the other platforms. I'm not really active on those other ones. So yeah, go ahead.
Yeah. Oh, yeah. So these, like a heterotroph is a microbe that likes the organic things. So if you think about like we are heterotrophs, our hamburger, or I always say hamburger, but like what hummus we eat is organic. And then autotroph means that it is it's like making its own food from CO2, like a plant does. So that's the difference between those things. And then so in each cases, the heterotrophic media, some of that we fed them was, which one was fun? Sometimes we grow cells. We grew Archiglobus and we like baked that Archiglobus and broke it apart into little pieces, and then we fed it itself. And then sometimes we just grow amino acids, right? So there's lots of
liquid, and then it's like the amino acids so that they know to eat.
Yeah.
Like our daughter, she works in a lab. She's studying the mind, and they know how to eat. So it's kind of confusing, but
yeah.
You ought to meet her. But anyway.
Yeah.
But I can understand things like that grow and stuff and live with.
Yeah. And you are right, like the mice just know how to eat, right? They just like see food and they're like, yeah, I eat that. Some of the reason we pick these foods is because when we looked in the genomes, this is just like a graph, a genomic map of one of the microbes I looked at. And I saw that they have these transporters that are designed to bring amino acids into the cell. And so it's like, oh, well, if they have that transporter, they must be transporting in amino acids, or maybe they're transferring them out, but maybe they're transferring them in. We also, like, this is just one organism, this hydrotherm archaeota, but in some of the others, we saw transporters for lipids. So then we kind of just have to guess, like, what lipid might it want? So I fed it, I put a bunch in, but it's tricky because maybe one lipid it wants to eat, and then, maybe another one it doesn't like. Right. Or maybe they're just like too overwhelmed and like, this is too much organics. I live in a place where there's not a lot of food, and now you're just kind of forcing food all around me, and I just can't deal with it. It's really tricky. That's why we really can only grow a very small percentage of the microbes, because it's tricky to get it just right. We're pretty good at growing microbes from the human body. We've had a lot of practice with that, but not so much in like weird environments, to know exactly what they want and at the concentrations they want without poisoning them. It's tricky.
So it's like a trial and error.
Yeah, exactly. Yeah, and I tell the students, like, it's like when you're cooking and someone has made you this recipe with 2 cups flour. Like, why? Like, why not 2 and 1/8 of a cup of flour, right? And like, so you make up this recipe, and they maybe like made it slightly off, and I'm like, how do you even know that was the right recipe to begin with? So let's just try this and see if it works.
Thank you. And once you guys, if you leave here and you have more questions, feel free to reach out. I do have a card of mine up there. I'm not sure if Amy put hers up there, but I can get you connected with our biology professor here, or we can provide you with contact information for Dr. Carr if you didn't catch that. So again, thank you all for coming and appreciate you being here.
Good time. Dang. Guess I'll just close. I think so.